class. Welcome to Chapter 3, where we're going to talk about protein structure and their function. So the actual sequence of the proteins, meaning the amino acids that make them up, specifies the folding into secondary and tertiary structures such as alpha helices and beta sheets, etc. And these are either functional units or can interact with other peptides to form quaternary structures. So multiple helices and beta sheets can form these larger structures with multiple subunits. And so exceptional conformational flexibilities of disordered proteins, contribute to their multiple function. So what that's saying is that different proteins can fold in different ways to have different functions. Some polypeptides with dissimilar sequences fold into similar 3D structures. We have these 3D structures, even though the amino acid sequence is different, these structures are very similar, like those alpha helices and beta sheets as we talked about. And then homologous proteins evolved from a common ancestor, have similar sequence, structures, and functions, and can be classified into families. And so globins in mice versus human have all evolved from similar genes, and thus can be classified into these families or superfamilies based on their structures. So what's interesting about proteins is there's only roughly 26,000 genes in the human genome, but there are hundreds of thousands of different proteins. And this is because there are alterations in splicing. So we have what's called alternative splicing, where different exons from a gene are incorporated together to make up different proteins. So one single gene can make a multitude of different proteins. And so looking at the structure of proteins, we kind of have this hierarchical structure where we have the primary sequence, which is the amino acids, the actual sequence of amino acids like lysine and proline and valine, etc. And then we have the secondary structure, which is the actual local folding of these amino acids, and this is where we form the alpha helices and the beta sheets. And then we have tertiary structure, where we get three-dimensional shapes. So secondary structure is only two-dimensional. Tertiary, we get this three-dimensional shape. And then we have quaternary structure, which is this multimeric structure, where we have multiple subunits, like hemoglobin's made up of multiple subunits, or histones, which make up chromatin, etc. And they come together to form these large complexes. And the same thing can be said for supramolecular complexes, which are these very large, tens of hundreds of subunit protein complexes. Now, tertiary proteins that don't associate into quaternary or supramolecular structures, they can have function, as well as quaternary or multi-subunit proteins or supramolecular proteins can all have function. Now, the function of protein kind of depends on their specific binding interactions and their conformational changes of the fully functional or fully developed protein. So structural proteins, they organize the genome, organelles, cytoplasm, protein complexes, and membranes in the three-dimensional space. So one example of this would be the histone proteins that make up chromatin to help give DNA its structure and its when it kind of binds it up. We have regulation, which is right here. So right there. So regulation is controlling other proteins' activity. We have signaling, which is monitoring the environment and transmitting information. So this could be enzymes that or proteins that sense the amount of lactose present to turn on the lac operon, things of that nature. Transport, so we have moving small molecules and ions across membranes, so like the sodium-potassium pumps that are on the cell membrane. And then we have enzymatic activity, so something to catalyze these chemical reactions. Remember from our last lecture, to reduce the amount of energy required to do some sort of chemical processing. It could be glycolysis or something of that nature. And lastly, we have motors, and these are proteins that generate movement. And so these could be little motor proteins that are walking along the actin cytoskeleton. They could be the motor proteins associated with cilia or flagella, etc. So as stated before, this is kind of a visual representation. So the primary structure of a protein is the actual sequence of the amino acids within that protein. The secondary structure is this local folding. So we have alpha helices and beta sheets, and these are kind of two-dimensional structures. Taking all the alpha helices and beta sheets into account and looking at a larger 3D structure, we get the tertiary structure. So this is for each subunit, the 3D structure of that protein. Then lastly, we have the quaternary structure. And the quaternary structure is made up of multiple tertiary structure subunits. And so this is like a macromolecule or macroprotein that has, in this case, 2 subunits. Examples of this would be hemoglobin, would be the histone proteins, would be hexokinase, things of that nature, where there's multiple subunits that make up one fully functional kind of macro protein. So just a reminder from last chapter, a protein is just a long chain of these amino acids. And these amino acids become linked through this dehydration reaction, which links a free amino end on the N-terminus to a free carboxyl end on the C-terminus. And so here, down in B, we can see that there is an amino end, which is this NH3, and this carboxyl end that has the C bound to double bound to an oxygen and single bound to another oxygen. And these peptide bonds, which are indicated in yellow down here, are the links between each adjacent amino acid. And so the function of these proteins depends on the sequence and the makeup of the protein sequence amino acids. So different combinations of different amino acids results in different amounts of folding and different conformations, and that gives the protein different structures, or excuse me, different functions. The structure gives different functions. So let's take a closer look at alpha helices here. So this is a very common secondary structures in proteins, and it's a very stable arrangement of polypeptides that are held together by these hydrogen bonds here that form between hydrogens and oxygens on the backbone of this amino acid sequence. And as you can see here, the R groups are all kind of facing outward. I should make my arrow go this way. And those are, remember, the functional parts of the amino acid that give each amino acid its unique characteristics and also react and bind to ligands and other proteins, etc. What's interesting and of note here is that proline, it can't participate in hydrogen bonding and therefore is usually excluded from alpha helices. So you won't see it in alpha helices. So another common secondary structure is the beta sheet. And these are laterally packed beta strands, each of which is nearly fully extended polypeptide segment. And so, as you can see here, we have this beta sheet segment, a loop, another beta sheet segment, and another loop. And what holds them together in this conformation, again, is these hydrogen bonds that occur between oxygens and hydrogens on each strand of the loop. And that's what stabilizes the structure. And in addition, just like the alpha helices, the R groups of this secondary structure are all facing outwards, as you can see here. And so the actual beta sheet itself is in the middle, and the functional R groups that give each amino acid its distinct characteristics are facing outwards. And also of note is that these beta sheets can form kind of two conformations. You can have anti-parallel, where you have a loop or a sheet going in one direction, it does a 180 and goes in the other direction, does a 180 and goes in the final direction. Or you can have a parallel beta sheet, where you have it go in one, the actual beta sheet segment go in one direction, then a long chain that does a 180 and another 180, and then again that beta sheet is in the same direction as the first beta sheet, and then a 180 and another 180, and then the third segment is also in the same direction as the first two segments. So those 180s that I talked about in the last slide are actually called beta turns. And so these consist of 4 residues, and they reverse direction of a polypeptide chain. They do that 180 turn. And these C alpha carbons of the first and fourth residue are usually thus less than 0.7 nanometers apart and linked by a hydrogen bond. So this one and this one are less than 0.7 nanometers apart, and they're linked by this hydrogen bond right here. And then the beta turn facilitates the folding of long polypeptides into this compact structure. And the R groups right here, these two, are usually glycine or proline, because of their glycine is the smallest of the R groups, and proline has this built-in bend, so they are most commonly found in these beta turns. So there's a model for how these proteins fold, and it's called the oil drop model of protein folding. So the tertiary structure is stabilized primarily by hydrophobic interactions between the nonpolar side chains and hydrogen bonds involving polar side chains, and the backbone amido and carboxyl groups. So what this is saying is that these hydrophobic residues, so we have the most hydrophobic as yellow here, as you can see in this kind of unwound amino acid chain or protein, they want to cluster together in the center because they're hydrophobic, so they want to be away from water. So if we look down on this one where we've kind of got a cross section of the protein, we can see that the inside has all these hydrophobic amino acids that don't want to be in the water. And so the charged and uncharged polar side chains, which are indicated as blue here, they want to interact with water and ions that are in the water. And so they want to be on the outside surface because of those charges that they have. They can interact with the polarity of water, and so they want to be on the outside. So the hydrophobic amino acids want to secrete or to hide in the inside, and the hydrophilic or the charged amino acids want to be on the outside. So you can kind of think of this similar to the phospholipid bilayer, where we have a polar head and the nonpolar tails, and the polar head wants to be on the outside associating with water, and then the hydrophobic or nonpolar tails want to be on the inside protected from water. So there are 5 ways that we can visualize protein structure. The first is this C-alpha backbone trace, which depicts how the polypeptide is tightly packed into a small volume. The second is a ball and stick representation, and this is similar, but it also reveals the locations of each atom as those little balls within there. We have a ribbon diagram, which emphasizes how beta strands and alpha helices, beta strands being blue and alpha helices being green in this case, are organized into the protein. We have a water accessible surface model, which just shows the topology of the surface, as well as the positive charges, which are blue, and the negative charged regions, which are red. And then lastly, we have a hybrid model that kind of overlays this ribbon structure with this water-based, water-accessible surface structure. We have a couple more motifs that are found within secondary structure, and these motifs generally give us an idea of what the function of a protein is. And so we have this coiled coil motif, which is where 2 alpha helices are wound around each other. And the seam stabilizes the interactions between the heptad 1 and 4 hydrophobic side chains. So the red and the blue here is stabilized, and they bind to each other at regular intervals along this chain. In addition, we have this helix loop helix motif. I've also heard it called helix turn helix motif, and that's where we have a helix and then the amino acid loop or structure or the amino acid chain will form this loop, and then we go into another helix, so we can get kind of a 90-degree turn in our protein structure. And lastly, we have this zinc finger motif. And so the zinc finger motif is common and very important in DNA binding proteins that help to regulate transcription. So this is kind of the active site for binding to DNA. And as you can see here, we have an alpha helices, and then this loop structure, but beyond this loop structure, we always have this zinc molecule bound within this motif. So here we have an example of one of these molecular machines that are comprised of a lot of different subunits, so this kind of quaternary structure. And this is the transcription initiation complex, and this contains tens to hundreds of polypeptide chains and sometimes other biopolymers such as nucleic acids. And so in this case, the transcription initiation complex has an RNA polymerase, so this is the actual enzyme that turns DNA into, or transcribes it into RNA, a mediator complex, which is made of roughly 20 subunits, and helps to assemble all these things at the gene promoter, so they mediate this process. And then all these other transcription factors that are responsible for initiation of transcription, but also they're kind of the on-off switch of transcription. So they're required to be turned on themselves upstream in order for this gene to be transcribed. For example, if you are going to do a lactase gene, a gene that is there to break down lactose, there might be some sort of mechanism to initiate the lac operon to express these lactose transcription factors, because if there's no lactose present, there's no point in making lactase to break it down. So those are kind of the on-off switch. They sense the environment to determine if transcription should proceed or not. So as you can see, all of these things, the RNA polymerase, the mediator complex, and all these transcription factors, they come together to form this transcription pre-initiation complex, which will allow RNA polymerase to then go and transcribe that gene into RNA and then later be translated into a protein. Let's talk briefly about protein folding here. So the amino acid sequence of the protein is what determines its 3D structure as well as its function. There's ATP-dependent molecular chaperones and chaperonins, and these are kind of protective proteins, such as barrel proteins, that these amino acid sequence will go and they will, the chaperones and chaperonins will help them fold into its proper conformation. And in the case where the chaperones or chaperonins don't work, or other environmental factors of the cell aren't ideal, then we can get misfolded amyloid fibril aggregates. And these are what accrue inside your brain that lead to the development of Alzheimer's disease, and a similar mechanism is present in Parkinson's disease. So planar peptide bonds limit the shape into which the protein can fold. So when there's a double bond, as exemplified between this carbon and this nitrogen here, there is kind of a 2D planar orientation of this section of the peptide chain. And so in this case, these bonds lead to two conformations. We have a trans peptide bond, and that's where we have the first amino acid group, or the first peptide bond on one side, and the second peptide bond on the opposite side. or we have a cis conformation where both peptide bonds are on the same side of this plane. And so in nature, 99% of the peptide bonds that have residues other than proline, so anything other than proline at this P2 bond, are in the trans conformation. So we see, as long as there's not a proline at P2, then we see this conformation. So if we were to look at this protein folding pathway in its entirety, we would start with this primary sequence, and in steps B, C, and D here, we would do the folding of our secondary structure. So first we would create these beta sheets and alpha helices, and they would kind of move around and get into position where finally we end up with E, which is our tertiary 3D structure. And the native state of this structure, so the kind of baseline, most common state of a protein is the conformation, the folded conformation that requires the lowest amount of energy. So whatever is the easiest for it to fold into, based on the energy, just standing state energy in the cell, is what is going to be the baseline most common conformation for that protein. So let's look at one of these molecular chaperone-mediated protein folding processes. So molecular chaperones bind to a short segment of a protein substrate and stabilize unfolded or partly folded proteins, preventing aggregation, so them sticking together, or degradation, them being torn apart. And so here we have an example with Hsp70 on the left-hand side here. Hsp is a heat shock protein that was initially observed in animals responding to increased heat. And so in step A, or step 1, we have heat shock protein binding unfolded protein to the open conformation of its substrate binding domain. So as you can see here, this orange segment is the substrate binding domain that binds the amino acid sequence, the unfolded protein. And then ATP comes in and it binds to the nucleotide binding domain, which is this light blue segment. So then, take a step back, because there needs to be energy input for this folding process. So in step 2, these co-chaperone accessory proteins, DNAJ and heat shock protein 40, with the addition of energy provided by that ATP, causes a conformational change in the substrate binding domain that locks the unfolded protein in place. So as you can see in this representation, now the orange substrate binding domain is now wrapped around that amino acid sequence and has locked it into place. Now GRPE and BAG1 come in and they take away the ADP. Remember, it gave its energy to change the substrate binding domain for a TP, and that will allow for the release of the protein and the return to the original state, and then we can go through the cycle one more time. Now let's look at a second type of chaperone folding. In this case we have Hsp, or heat shock protein 90. So in step 1 there is this very flexible dimer that has an open configuration that can bind a client or an unfolded protein. So we have 2 of these nucleotide binding domains that are indicated in the light blue here. And so in step 2, ATP comes in and binds to the nucleotide binding domains and causes a very rapid conformational change that brings the domains closer together to dimerize, and the substrate binding domain also moves together, and this is when the amino acid chain moves in. And from there, we get that rapid partial movement, but then there is a slow movement to a fully enclosed inner state of the protein. After this closed conformation is achieved and the client protein is being folded, then we have another conformational change where ATP is converted to ADP, releasing energy, and that causes this protein to be released after the folding. So this picture isn't shown of the actual releasing, but we get a released state here. ADP then is released, and we go back to this open flexible conformation, and another protein can come in and be folded. So these cycles are continuous, and these chaperone proteins can fold multiple different amino acid chains into their final conformation. So now let's talk here about the chaperonin mediated protein folding. So chaperonins are these folding chambers in which all or part of an unfolded protein can be bound in an appropriate environment, giving it time to fold properly. So these are kind of like little shelters that help to fold the protein outside or in a confined area, so there's not outside influences that could lead to improper folding. So we'll talk here about GROL, which is a prokaryotic chaperonin. And so these are these barrel-shaped complexes. Throughout this chapter I've been talking about barrel proteins, so this is an example. So these are barrel-shaped complexes of 14 identical subunits arranged in 2 stacks or rings. And each of these rings, so we have the top and we have the bottom, each of these rings is 7 subunits that form these kind of barrel-shaped chambers. And on top of that, there are these GRO-S as opposed to GRO-L lids that will seal the top and the bottom of the chambers. So let's look at the cycle here. So in step 1, a partially folded, or this is important, a misfolded protein. So if a protein isn't folded the way it should, you can go through this process as well. So a folded or unfolded or misfolded protein enters the folding chamber right here. And then the second chamber, the bottom chamber, is blocked by one of those GRO-S lids. In step 2, each ring of the 7 GRO-L subunits, so remember there's 7 for each of those 2 chambers, they hydrolyze an ATP and release ADPs in a set order which coordinate the GRO-S binding and release and the polypeptide binding, folding, and release. So that's where the energy comes in. Each one of those 7 subunits will hydrolyze ATP and allow energy to allow these folding and conformational changes. So in step 3, the polypeptides remain encased in the chamber, which is capped by the lid, where it can undergo folding until the hydrolysis, which is the slowest step in the cycle, induces binding of ATP and a different GRO-S on the other ring. So it will move the lid to the top. So this is incorrect right here. The lid should be, oh no, I'm sorry, that is correct. The lid at this stage gets moved to the bottom. Then we have in step 4 the release of the lid as well as the bound ADP, and this allows the newly folded protein to diffuse from the chamber and go about its business and go and do its biological function. Now, if for some reason this protein was improperly or incompletely folded or misfolded, then it can enter back into another chamber and be fixed. And so this cycle will continue until the protein is in its proper conformation. So we talked briefly earlier about this cis-trans conformation across peptide bonds. And so in these planar double-bound peptide bonds, there is a 99.97% chance that this P2 is in a trans conformation. And that is the case for all peptides except for proline. So for proline, however, about 5% of peptide bonds are in the cis conformation. So they are on the same side of this double bond, if we were to draw it like that. And proline isomerases are an enzyme that catalyze the cis-trans isomerization to facilitate this protein folding. And so depending on whether this proline is in cis or trans isomer form, it alters the structure of a protein. And so as you can see here, we have a trans conformation at proline number 287 of the amino acid sequence. And here we have a cis conformation of proline 287. And you can see we have changes in the structure based on that proline's cis or trans conformation. And so this stereochemistry of the amino acids can influence the protein's activity. So you have different isomers or different forms of this protein. Even though the amino acid sequence is exactly the same, depending on the stereochemistry, whether they're cis or trans, we can change the activity of that enzyme. And that proline isomerase, that catalyst that can change conformations, may act as a switch that regulates this protein activity by flipping between cis and trans. So misfolded proteins or proteolytic fragments, so pieces of proteins, can accumulate as aggregates or plaques inside or outside cells. And so this leads to some disorders and diseases. And these aggregates can occur between bones, within the liver, and also within the brain. And so there's a lot of different proteins that can aggregate into these amyloid, well-ordered fibrils. And they have a common structure that can cause amyloidosis disease, which includes neurodegenerative diseases such as Alzheimer's and Parkinson's, as we talked about earlier. And it also causes mad cow disease in cows and sheep. And so what happens is, let me get my cursor here, there are these sheets of either fragments of a protein or a misfolded protein, and they hydrogen bond in between them and form these long fibrils, which are essentially beta sheet fibrils, and they just stack on top of each other, one on top of another, and form these long strands. And then these long strands then kind of wrap around each other, as you can see here, where you have multiple strands forming, and then kind of like a thread twisting around one another. And these sheets pack real closely together to form what is called a protofilament. And these protofilaments assemble into thicker fibrils, so just like thread or rope, you have small wraps together, and then those filaments will wrap into a larger fiber. And these fibers will aggregate into these macroscopic plaques. And so if we were to look at under a microscope some brain tissue of an Alzheimer's patient, what we would see are these long fibrous plaques that have accumulated in these patients, and that's what leads to the phenotypes of Alzheimer's disease. So let's talk a little bit here about protein binding and how enzymes work and catalysis. Protein function depends on binding other molecules or ligands. This is evident because they need to actually interact with something to actually have a function. In the case of enzymes, they accelerate rates of cellular reactions by lowering the activation energy and stabilizing transition state intermediates. Enzymes often use acid-base catalysis, which is mediated by one or more amino acid side chains, and then metabolic pathway enzymes may be associated as domains of monomeric protein subunits or subunits of a multimeric protein or components of a protein complex assembled on a common scaffold. So we'll get into some examples of these structures and functions. First, let's look at protein ligand binding of antibodies. And remember, a ligand is a molecule to which a protein binds. So it's anything that a protein is looking to bind to. So in this example, we have an IgG antibody, and this is represented in the ribbon model here. And we have two identical heavy chains that are in the red here, circled right there, and two identical light chains, which are in the blue here, and there's the other heavy chain. And these are linked covalently by disulfide bonds. So the CDR, the heavy red over here and over here, is the complementarity determining regions. And this is composed of 6 highly variable loops which form the antigen binding sites. And the variability makes them specific to different antigens. And looking down here, this is kind of a stick drawing of the protein, where we have the heavy and the light regions. The CDR, they refer to it as a hand-in-glove fit, and this means that there's molecular complementarity between the antibiotic CDR and the site to which it binds on its antigen. And so this shape determined by those changes in loops in the CDR is specific for the antigen that it's going to bind to. And as you can see here, it forms this nice tight bond to the antigen, and then it can perform its function. So now we'll look at another enzyme. In this case, we have trypsin. And these enzymes, such as trypsin, catalyze making or breaking the covalent bonds found in a substrate. And so here, trypsin, otherwise known as the serine protease, has an active site. And within this active site, we have this substrate binding site, which is indicated in blue here. And we also have a catalytic site, and that is indicated in the violet or purple here. And in this binding site, there's a side chain of these, what we call a catalytic triad. And there are 3 amino acids, serine 195, aspartate 102, and histidine 57. And these 3 work together to break apart peptide bonds. And so this is also shown here in the ribbon model, where we have the binding site and the catalytic site. And they didn't indicate which color is which, but you can see this purple, this blue, and this red amino acid, which are the ones that have the catalytic action. In some enzymes, the catalytic and substrate binding sites overlap or are very close to each other, such as in this case here. You see they're right next to each other. But in other enzymes, the 2 regions can be structurally distant. So here we just have that same schematic from the last slide, but this is kind of showing how the reaction mechanism works. And so serine proteases digest meals, so they're pancreatic enzymes like trypsin, chymotrypsin, and elastase, but they also control blood clotting and help silk moths chew their way through their cocoons. So they have an enzyme, a really cool enzyme called cocoonase, which is a great name. And so the enzyme substrate binding site and the catalytic site cooperate in this multi-step reaction to convert substrates to products. And so the enzyme, which we have indicated by E here, will bind the substrate molecule at this fixed activity or active site. When they're together, we call this the enzyme substrate complex because they're bound into one chunk at this point. And as the enzyme is performing its function, they convert this, what was first called the substrate, into what we now call products. So we'll have two products in this case because we have broken this substrate into two pieces. So let's look at this trypsin binding a little more closely here. So the active site, which is indicated on A here, the substrate will form two stranded beta sheets with the trypsin substrate binding site. So we have one sheet here, one sheet there, and we have these hydrogen bonds kind of holding it in place. In this side pocket, which is right over here, that will bind the substrate arginine, the R3 side chain. So this is the arginine R3 side chain right there. Down below it we have Hsp190, which has a negative charge and stabilizes this arginine in place because since it's negative charged, and this arginine guanidinium group here is positively charged. It's kind of like magnets, right? So they'll hold them in place there. And the binding allows the substrate arginine peptide bond up here for hydrolysis catalyzed by that enzyme's active site catalytic triad. So it's catalyzed by these three right there. So in the previous slide we talked about how there are differences in these enzymes in your stomach based on what they break apart. And so in the first case here, for trypsin, we have a pocket that binds positively charged arginine and lysine side chains. In chymotrypsin, it binds large hydrophobic side chains such as phenylalanine. And then in the last case, elastase, it binds small side chains such as glycine and alanine. So what makes these enzymes different is their pockets and the active amino acids involved in binding these side chains to allow that triad to perform the cleavage or hydrolysis. So when we have these multi-enzyme complexes, sometimes there can be issues with efficiency. And so there is a way to fix this, and that is by incorporating a scaffold protein. And so if we have, say we have three enzymes that are required for processing a reactant to a product. In this case we have A, B, and C. Now if they're just free floating in the cytoplasm waiting for A to convert to an intermediate product, and then it drift over to B to convert into another intermediate product, and then find enzyme C floating around to convert into a product, this is done by free diffusion, right? They're just floating around, and you have to come into contact randomly with the right intermediate product and the right enzyme, etc. And so it can take time, and it's not very efficient. So one way to fix this is either to form these scaffold multi-enzyme complexes. So the scaffold's just a protein that holds them all very near each other, these enzymes that are actually doing the work. And so when A processes a reactant and makes an intermediate, it goes right to B because it's right there, and then it goes right to C because it's right there, and then it ejects your end product. Or there can be these multi-unit enzymes that bind to one another and can kind of, they don't really need a scaffold protein, they just have domains on them that allow them to stay in close contact to one another. Another possibility is that instead of having these multi-protein macromolecules, we could have a genetically linked macromolecule. In this case, the genes or these enzymes are fused at the genetic level, and that means that one gene encodes all A, B, and C in this case, and so the polypeptide chains are created as a single polypeptide chain, and it folds into this single protein with 3 subdomains, A, B, and C, and thus when this processing of reactants to products occurs, all 3 subunits that make these different intermediate products, they are all in close proximity, and that speeds up the reaction speed. So now we'll move on to regulation of protein. So proteins may be regulated at the level of the protein synthesis. It can be regulated by degrading proteins that already are there, or through non-covalent or covalent interactions. Proteins marked for destruction with a ubiquitin or polyubiquitin tag by ubiquitin ligase are degraded in the proteasomes. There are several allosteric mechanisms that act as switches, reversibly turning protein activity on and off. And then there are higher order regulations or regulators, which includes the intracellular compartmentalization of protease. So ubiquitin and the proteasome are responsible for proteolysis, which is the breaking down of proteins. So the proteasome has this distinct shape where there's this 20S core where there is an alpha, beta, beta, then alpha ring. And these inner chambers are where the actual breakdown of the protein occurs. On either side of this chamber, we have 2 19S caps, which is comprised of 19 subunits, where the 19S comes from. There are 2 ubiquitin receptors that sense the ubiquitin on the targeted protein that is to be broken down. There is a dub, which is the D-ubiquitin site, which removes the ubiquitin, and then also an ATPase site, which breaks down the ATP required for the folding or the hydrolysis of the protein. So let's look at the steps here. So in step 1 down at the bottom here, we have an enzyme E1, which is activated by attachment of a ubiquitin molecule. E1 then transfers the ubiquitin molecule to a cytosine residue on E2, which is the ubiquitin conjugating enzyme. Ubiquitin ligase E3 transfers the E2 bound ubiquitin molecule to the side chain of a lysine residue in a targeted protein. So this E3 adds the ubiquitin as a tag onto the protein that is to be targeted. E3 adds ubiquitin molecules to the previously added ubiquitin on the target protein. So E3 will then add more ubiquitin molecules to the previous ubiquitin molecule. So you have this chain of ubiquitins. So we have our protein that we're going to break down, and now we've added a bunch of ubiquitins in a chain to it. So during recognition, the proteasome's 19S cap, the UB receptors bind to this polyubiquitinated tail, or this long chain of ubiquitins. So you can see here that we need multiple this long chain because we need to reach both of those 2 ubiquitin receptors. During release, the D-ubiquitinase enzyme removes the ubiquitin groups, and then we have, that's right here, D-ubiquitination. Then we have unfolding, where ATP hydrolysis enables the 6-protein hexameric ATPase subunits. So those were the inner chambers that we talked about before, to unfold the substrate. So at that point, it is it's happening in the ATPase, right up here in the red, is unfolded, and then it moves into the inner chamber. And at the inner chamber, peptide bonds are cleaved, and the cleaved peptides are then discharged and degraded further by soluble proteases that are floating around in the cytoplasm. So another example of an enzyme being activated is the conformational changes of calmodulin. So this is an allosteric switch, so this means that there is a ligand or an allosteric effector binding to an allosteric site. And so calmodulin is this widely distributed cytosolic protein, and it has these 4 EF hand domains. And so EF hands are that helix loop helix motif that we had talked about earlier. And so when there are low cytosolic calcium concentrations, so less than 5 × 10⁻⁷ molar, so very, very small, we have this unbound dumbbell conformation. So you can kind of see, they call it a dumbbell because it kind of looks like that. When the cytosolic concentrations of calcium increase to over 5 × 10⁻⁷, the calcium binding changes the calmodulin conformation. So we see that those EF hands will bind up to calcium when there's a high concentration, so those 4 sites right there. And what this does is it brings the hydrophobic side chains and it makes them more exposed to the solvent. And this calmodulin complex will then wrap around this target peptide. Specifically, it will wrap around a helices of a target peptide, and it will then alter the function or activity of that target peptide. So calmodulin senses changes in the cytoplasmic calcium. It binds that calcium and changes conformation, and then tells another enzyme or protein that it needs to behave differently. Okay, so let's talk a little bit about this GTPase switch. So GTPases are this superfamily of proteins that exist in 2 conformations, this GTP bound or active or on conformation, and this GDP inactive or off conformation. And so these GTPases, they hydrolyze GTP to GDP. And in the GTP bound form, the active or on form, this can interact with other target proteins somewhere else. And well, this will be our target protein. And they can then regulate the activity of that protein. So they can turn it on, enhance it, etc. And so the GDP bound protein is inactive. And so this down here is not able to go and regulate other proteins because it's in this off conformation. But there are a couple other cofactors here that are able to allow GDP to become the active form and the active form to become the inactive form. And so guanine nucleotide exchange factor, it will remove GDP from the inactive GTPase and it will add GTP. So we're removing GDP, adding GTP to make it active. And this GTPase activating protein, what that will do is it will allow this GTP to donate this phosphate group down here, which generates energy, and this happens when it goes and interacts with other proteins to regulate them. And that will cause this GTPase to become inactive because now, since that GTP donated its phosphate group, we now have GDP. So you can see this is it can be recharged and by GEF, and then when it needs to expend energy to regulate another protein, this GTPase activating protein or GAP will allow it to donate a phosphate group, which will generate energy, and then it's inactive and it goes round and round. So when we talk about activating or inactivating a protein, there are really 2 main players or families of players, I should say, that do this. And so we'll start with kinases. So a protein kinase is an enzyme that takes ATP and it takes off one of those phosphate groups and makes ADP. But then what it does is it adds that phosphate group to a target protein. And so this target protein can either be turned on or it can be turned off by that, depending on the protein. So what this is doing is making a slight conformational change to the protein, so it's either inactivating it or activating it. Now, of course, this is a cycle just like before. And so protein phosphatase, what that does is it takes water in a hydrolysis reaction and it removes that phosphate group that was added to the target protein to activate it or inactivate it. And then that, in turn, will either activate or deactivate a protein. So if adding this phosphate group to the target protein inactivated it, removing it will turn it active, or like the slide says, if the kinase activity activated a protein, removing that phosphate group with a phosphatase will inactivate it. So it's all very dependent on the protein that you're actually targeting. So in the human genome, I believe there's over 500 kinases that regulate different proteins. They can regulate multiple proteins or a single protein. So this is a giant family that goes and turns proteins on, turns proteins off. So let's look at briefly look at how we study proteins. And so proteins can be isolated from other cellular components based on a variety of physical or chemical properties. They can be quantified by various assays and more specifically antibody recognition. And tagging these various types of markers can be used to investigate the synthesis of the protein, the cellular location, whether they're nuclear, membrane proteins, organelle proteins, etc. Processing, where they go within the cell, if they start in the nucleus and then they leave, or if they start in the cytoplasm and go to the membrane, etc. And their stability, how long they're there, so we can tag them and look at how long that tag exists, because once that protein gets these ubiquitin markers to go to be degraded, then that marker will be degraded too, so we can see how long it lasts in the cell. X-ray crystallography, cryo-electron microscopy, and NMR spectroscopy will give us those 3D structures that we've looked at earlier, the ribbon structures and things like that, in addition to a lot of modeling with proteomics and looking at the sequence. We can kind of predict what these structures are. So I'm going to quickly hit on this because this is probably something familiar to you from your chemistry labs, if you've taken biochemistry, general chemistry, or some biology labs, micro, etc. We can separate particles based on centrifugation, right? So heavier stuff falls to the bottom first, very light stuff will stay in the supernatant unless you centrifuge it for a very long time. And so what you can do here is centrifuge for a amount of time specific to what protein you're looking for. So if you're looking for a very heavy protein, a supermolecular complex like a hemoglobin or something that's pretty big, ribisco, things of that nature, you can centrifuge it very shortly and the heavy stuff falls to the bottom. You can get rid of the supernatant, which will have all the other proteins, and then just collect the pellet, the heavy stuff, and your protein should be in there. You can also do a sucrose gradient, and this will help to provide resolution. So if you create a sucrose gradient where there's high concentrations of sucrose at the bottom and low concentrations at the top, you have a changing density throughout your centrifuge tube. And so you can increase the resolution because things will settle out into these bands, and then you can collect those fractions and look for your protein in one of those fractions based on what your predicted molecular mass of the particle is, or of the protein is. So another technique that can be used to study proteins is SDS-PAGE, which is a polyacrylamide gel. and this will separate proteins primarily based on their mass. And so in step 1, what happens is proteins are treated with SDS, which is a negatively charged detergent, and that interacts with the hydrophobic amino acids to dissociate these multi-America proteins and denature the protein structure. And when it does that, it also coats each protein with these negatively charged ions that are proportional to the mass of the protein. So we're breaking this big structure apart and we're making them linear and getting rid of that 3D structure and then coating it with charges. And the number of charges are associated with how big the protein is, or the mass of the protein. And then we do electrophoresis. So this electric field will drive these SDS protein complexes that have been denatured through this polyacrylamide gel. And small complexes move through the pores faster than large complexes. This is very similar to running a gel for DNA. And the proteins separate into bands according to their size as they migrate. And then the separated protein bands are stained with a dye, and we can visualize that where these proteins are within the gel. So as you can see down at the bottom here, we have large proteins don't migrate that far because they have a harder time getting through the pores, while small proteins migrate much further. And then if we use a ladder that we have known bands of known size, we can measure the size of that protein. And if we're trying to isolate it, we can cut out this little band, get rid of the polyacrylamide gel, and then have our protein isolated. Now, if we have proteins that we can't get accurate resolution because there are a lot of other proteins of similar size or an isomer that is very similar, what we can do is we can do 2D gel electrophoresis. And so the way that this works is the first step is to do this isoelectric focusing. And this, instead of separating based on their charge, this separates proteins, based on their size. This separates proteins based on their isoelectric point, or their net charge. So depending on their amino acid concentration, or what their makeup is of amino acids, they'll have different amounts of positive and negatively charged amino acids. And so they will move along a pH gradient, which we see here, a pH from 4 to 10, until they find a place where the pH causes their net charge to be 0. And then once we've resolved all these proteins based on their charge, we flip the gel 90 degrees, and then we perform just the regular SDS-PAGE electrophoresis. And so in one direction, we have our pH gradient, and then in the other direction, 90 degrees, we have their molecular weight gradient. So as you can see here, if we have, for example, this protein right here, and this protein right below it, if we were trying to do that just based on the molecular weight, they would move together because they both have similar molecular weights. They both have similar molecular charges. So if we were only doing the isoelectric focusing, we won't be able to resolve them. Similarly, if we were to look at this second protein here and this protein over here, and we were to do only molecular weight, we won't be able to distinguish them either because they're parallel to each other, right? They're on the same line. And so we wouldn't know if they are our protein or a different protein. But by doing both of these methods of electrophoresis for molecular weight and isoelectric focusing, we can resolve these proteins that are similar size, or we can resolve proteins that are similar charge to really get more specific to find our protein of interest. So another way that we can separate proteins is through chromatography. And so this is these are little columns that have beads in them that we can use the characteristics of our protein in coordination with these beads to separate them. So the first is gel filtration chromatography, and this separates proteins just based on size. And so within the column, we have these porous beads, and within the beads are these little tiny holes. And so small proteins will start to diffuse through those holes, and it'll take them longer because they're going in and out of those small spaces, while large proteins will elute out of the supernatant sooner because they are not able to get in those holes, and so they just fall through the column pretty quickly. Now, the second method is this ion exchange chromatography, and the way that this works is we put beads that have a negative or positive charge on them. And so if we're looking at a negatively charged protein, or if we just want to look at negatively charged proteins, we'll put positively charged beads in the tube. And as this solution moves through, all the opposite charged proteins will adhere to the beads, while all the same charge, so if we have positive beads, all the positively charged proteins will be repelled and they'll move through the column much quicker. And then once we have all of our negatively charged proteins stuck to the beads, we can release them by adding a simple salt concentration that will go and it will take up those bonds with the beads and release our proteins, and then what we should have left is just our negatively charged proteins. So another way that we can characterize and look at proteins in the cell is through radiolabeling them, and this is using a radioactive isotope of a common element that will be incorporated into a protein and give off this radioactive signal that we can measure and then determine where it's located, how long it lives, etc. And so these are some of the common isotopes that are used. Phosphorus 32, which has a half-life of 14 1/3 days, iodine 125, sulfur 35, tritium, and carbon. And so depending on what we're studying, we can use different elements. So for example, phosphorus, that is not present in carbohydrates, but it's present in DNA, we could label with phosphorus, or sulfur, which is present in proteins, but it's not present in DNA. If we want to look at just proteins, we could label with sulfur. So depending on what we want to study, we can use these different radioisotopes. And so in the next slide, we'll get into an example of how this is done. So this example is called a pulse chase experiment, and it tracks pathways of a protein as they're modified within the cell. And so this is particularly useful for locating where the protein is, whether it's in the membrane or the nuclear membrane or an organelle, etc., or how it's modified over time. And so the pulse phase of this is where we incubate cells with, in this case, 35S methionine, which is a sulfur radiolabeled, and we incubate them for a half hour. And this allows all newly synthesized proteins that we're interested in to incorporate that radioactive sulfur as opposed to just a normal sulfur. So we'll be able to see all the newly synthesized proteins because they're radioactive. And so after that pulse of a half hour, we wash away all these amino acids that are radioactive. And so we reincorporate non-radioactive amino acids. And so then we incubate the cells for varying times, up to 24 hours, with that unlabeled methionine. So here they did a half hour, 1, 2, 4, 6, 8, 12, and 24 hours with non-radioactive methionine. And then we can immunoprecipitate and isolate one specific protein. In the example that we're looking at here, they looked at a low-density lipoprotein receptor. And so after that, SDS-PAGE of these precipitates is followed, and then we use auto radiography to look at the radioactive proteins. And so all that being said, let's look at the results, because that's a lot to digest, I'm sure. So the target protein was initially synthesized as a small precursor. So this lipoprotein was originally this precursor P. and then it's really rapidly modified to a larger, more mature form that we're calling M. So you see that 0 hours, there's this amount of P, and then it's really quickly turned into M and proceeds to exist up to about 24 hours where it starts to be degraded. Now, and as you can see here, the P is gone at a half hour. So it's really quickly turned into M and then exists as M. And so it's the precursor. However, in our mutant population, so we're looking at if you mutate this precursor P protein, what happens? And so in this mutated variety, we see that at 0 hours, we have a bunch of this precursor, but because it's mutated, it's never able to turn into M. And we see that P exists, but M is never made. And so using this radio-labeled sulfur, we're able to see the fate of P when it has a mutation in it, and that it's no longer converted into M. And so if you were to think of this from a medical standpoint, if you find a mutation and you want to, that's associated with a disease, and you want to know why that mutation is causing this disease, a pulse chase experiment can show you if there is some sort of interaction or interruption, for that matter, in the pathway that is causing this phenotype of this disease to occur.